How cells survive, ATP and the electron transport chain. In this video and its sequel, we'll explore cellular energy management. In reverse, starting with how cells survive and tracing each step back to putting food in your mouth. To guide us, we'll answer 6 key questions, starting with question 1, how do cells survive? Cells stay alive by doing 3 kinds of work. First, they carry out transport work. That's actively shuttling molecules across membranes against their concentration gradients. Second, they perform chemical work. That's building new molecules by forming chemical bonds. Third, they do mechanical work, generating force with motor proteins, contracting muscles, moving vesicles, or spinning bacterial flagella. All of these tasks require an input of energy. If a process requires energy, we call it endergonic. Endo means entering, erg means work, and is the same erg as the seen in the word energy. In living cells, that energy most often comes from hydrolyzing ATP, which releases the free energy needed to power these reactions. Question 2. How then do cells generate ATP? 2 ways: substrate-level phosphorylation and oxidative phosphorylation. In substrate-level phosphorylation, an enzyme transfers a phosphate from a high-energy substrate to ADP, but most ATP comes from oxidative phosphorylation via ATP synthase, a channel enzyme that literally cycles through 3 steps. Yes, it really spins. First, it grabs ADP and inorganic phosphate. Second, it forges them into ATP. Third, it releases the ATP so it can start the cycle again. Question 3. So, what causes ATP synthase to spin? ATP synthase is embedded in the mitochondria's inner membrane. Protons from the intermembrane space flow down their concentration gradient through ATP synthase's channel, driving its rotation, just like water turning a turbine in a dam. This proton gradient is called the proton motive force because it motivates ATP synthase to turn. But if protons simply flow down the gradient, it'll collapse in seconds. So, question 4, how is the proton gradient replenished? Enter the electron transport chain, or ETC. Literally a chain of protein complexes that transport electrons. And the word complex isn't fancy, it's just many proteins in one structure. Like an apartment complex is many apartments. We could think of the ETC as a bunch of apartment complexes, but to understand their function, I prefer to think of them as hungry little monsters. Each step of the ETC moves electrons from less electron-hungry to more electron-hungry complexes. Because these exchanges always move toward more stable, satisfied states, a bit of energy is released each time. Finally, oxygen, hungriest for electrons, snatches them, combines with protons, and forms H2O, about 1/3 of a liter of water per day in an adult. The energy released with each electron transfer is used to pump protons back into the intermembrane space, replenishing the gradient. Question 5. Where does the electron transport chain get electrons? 2 main carriers, NADH and FADH2, donate electrons. NADH gets extra attention because it donates earlier in the chain, so it drives more proton pumping, and about 5 times as much NADH is produced as FADH2. Think of our cell's pool of NAD plus and FAD as a fleet of empty trucks, and NADH and FADH2 are loaded ones. The NAD plus and FAD are loaded, bring their cargo to the ETC, and then unload those electrons, returning to their NAD plus and FAD forms. Anytime an electron moves from one molecule to another, it's a redox reaction. One molecule is oxidized, losing electrons, and the other is reduced, gaining them. We've already watched plenty of these transfers, but we haven't yet shown how NAD plus and FAD become NADH and FADH2. Which brings us to question 6. How are these electron carriers reduced? It's a big topic, so let's take a short break. When we return, we'll dive into central metabolism.